Okay, it looks like it's pretty close to time, right? I'm going to go ahead and introduce our speakers for today. Today we have Julia Warrick and Jordan Dopp, and they are both seniors, and they're working on a presentation about what they're doing in the lab this summer. So this summer they worked in the lab, which, if I put a plug in here, is amazing that undergraduates get to work in a lab like that. Our school allows that and some other schools don't, but they did amazing work over the summer and they're going to tell you about it. And then afterwards, there's going to be a code that pops up in their presentation. It's a QR code, and everyone who watches the presentation needs to go ahead and scan that and then answer those survey questions. Okay? So without further ado, I'm going to turn it over to your presenters.
Okay, so hi everyone. Like Lindsay said, working in Dr. Gibson's lab really is amazing because you're working with graduate level stuff even as an undergraduate. So it really does prepare you for life after Maryville College. And to also put in another plug, she has done great work to collaborate with other labs across the world, really, with this lab led by Dr. Valence Washington at the University of Puerto Rico. So some of the stuff we've done, he's kind of put in some input and helped us out with it. But this summer, we really worked to develop and optimize methods to characterize heparin binding and interactions with different proteins in the human body. Ooh. And to just give you some background on us, hi, my name is Jordan. I use she, her pronouns. I am a senior here studying biochemistry. I'm also a Bonner Scholar, so I do some community service. I'm a student ambassador, one of the Monday STEM Success Tutors, a lead academic mentor in the Academic Support Center, and I am involved with ACS and Tri Beta.
Hi, I'm Julia Warrick, and I'm also a senior and a biochemistry major with a psychology minor. And I'm a Scott Science Scholar, and I was also a peer mentor for S Cubed. And then I also tutor in the STEM Success Center, and I'm an SI leader and lab TA for biochemistry. So I started working in the lab my sophomore year when another student who was working in the lab, it was Boomer, he's here, so he reached out to me and asked if I'd be interested in doing lab work. So at first it was a lot of simple tasks like training us how to use pipettes and how to load gels, and then we were slowly given more projects and more independence in the lab. So if you're interested in ever working in a lab, don't be shy to reach out to us or other students who are working in different labs, because there's definitely always opportunities to do that.
Yeah, and so the experience was really the same for me. We were both friends with Boomer, and he was like, hey, there's this really cool opportunity. If you want to get some lab experiences, come check it out. And it was really nice getting to work with Dr. Gibson because it was a safe space to mess up. And it still is a safe space to mess up, even as seniors. But it was just cool to see how we came into the lab, barely being able to use a pipette, barely being able to pour a gel. And now, you know, here we are finishing up our senior thesis, about to graduate, and just to really like look back at where we've been and like where we are now. And it's great things for going to MC and beyond.
So this summer, I was awarded a Leyford Scholarship to work on a project that studied the interactions of the protein TL21 with immobilized heparin, which is just my senior thesis. And this scholarship was really nice because it provided us with money to get a lot of new supplies that we wouldn't have been able to get otherwise. And it was also really nice because that I got a stipend for working a certain number of hours over the summer. So it was really nice to have a stable full-time job during all of the craziness of COVID. And it also contributed to my requirements for Maryville and also to my future career. So that was really good. And I was also really glad to have gotten this scholarship to work on a project full-time over the summer, because I felt like it was kind of a way to get a glimpse of what grad school life would be like, or the research life beyond that. So I didn't, get to grad school and be like, oh, I actually hate working in the lab for 8 hours a day, because that really wouldn't be great. And I think it also just let me expand my skills a lot more and get more comfortable designing my own experiments and just being in the lab more often.
So for me and Julie are kind of the same person at this point. Just ditto to everything that she said. Instead of the Leyford, I did receive some money from the college through a senior study grant to buy some of the supplies for my project, which was similar to Julia's, but instead of looking at heparin immobilized to a surface, I looked at interactions of different proteins with heparin in solution. And I also employed some of my organic chemistry skills to work on some synthesis projects. And for me, it also did the same thing. It just helped provide some stability and a constant during all of the crazy things that are COVID-19. And now that you've gotten some background on us, we'd like to give you some background information on what we've been studying.
So heparin is a commonly used anticoagulant, and it has a lot of medical uses. It's commonly used to prevent CVAs, which are strokes, and then also to prevent heart attacks. And it just prevents those dangerous blood clots from forming. And then it's also used after those events happen. And then it's also used like after major surgery or several other things. And heparin is a glycosaminoglycan, and it has a lot of sulfate groups on it, so that makes it a really negatively charged molecule, and that contributes to how it's able to interact with other molecules in the body. And then fibrinogen is a major plasma protein, and it's about 340 kilodaltons in size, and it plays a major role in the coagulation cascade during the formation of blood clots. So fibrinogen is cleaved by thrombin in the final stage of the cascade, and it's cleaved into its active form known as fibrin, and that's actually what forms the meshwork of a blood clot. So in order for fibrinogen to do all of its functions during coagulation, it has to interact with several other proteins and molecules. So you can see on the figure that there are some highlighted amino acids, and that amino acid is arginine, and it's positively charged. So that allows fibrinogen to interact with other negatively charged molecules like heparin, and that is really important for fibrinogen's function. And then TL2-1 is called trim-like transcript 1, and it is a small protein that's found in platelets, and it has several different domains. There's a soluble form, and there's also a membrane-bound portion, but we just want to focus on the soluble form, really. So the soluble form of TL2-1 is released from the surface of platelets when they're activated during the coagulation cascade. And the TL2-1 goes and interacts with several other proteins and molecules, and it facilitates a lot of the inflammation that happens during coagulation. And so you can see there are more highlighted amino acids, and the blue are more arginines, and then the greens are lysines, and those are both positively charged. So they also facilitate interactions of TL2-1 in the same way that they did with fibrinogen. And so since TL2-1 has that strip of positively charged amino acids that you can see there, we think that those interact with the negatively charged sulfate groups of heparin, and that allows them to bind.
So we've given you a lot of information, but kind of putting it into context, the big so what of all of this is that heparin, when it's used medically, it can do a lot of good, right? It can reduce blood clot formations with people with strokes and heart attacks, but it can also do a lot of bad. And when it does bad, we call it heparin adverse events. One of those being heparin-induced thrombocytopenia, or HIT, or HIT, however you want to say it. And that's just a fancy word for low platelet counts after having heparin. And that's not good because it can put you at risk for different inflammatory diseases and just a lot of yucky stuff. So in an effort to improve heparin's medical use and maybe even some diagnostic testing for inflammatory diseases, it's really valuable to study heparin's molecular interactions with different proteins in the human body. And that's exactly what we've done. We've tried to develop and optimize different methods to study those interactions. And that's exactly what we're talking about with you today.
So this is some of the data that I collected over the summer. These are called ELISAs, and it's from a plate assay that I did using heparin-coated plates that we were actually able to get with the money from the Leyford Scholarship. So panel A is showing a graph that has the protein concentration on the x-axis and the average absorbance on the y. So the absorbance is basically just how much light the sample in a plate was able to absorb. And so we interpret that as how tightly the protein was able to bind to the heparin that was stuck to the plate. So the higher the absorbance, the tighter that it bound. And you can see that the dark blue line on top and the green lines, they correspond to TLT1. And those had a lot higher absorbances than the light blue and orange lines, which represent fibrinogen. So on this specific plate, fibrinogen didn't really bind, and we tried it multiple times and it just never developed. So this could be because the heparin is stuck to the plate, which means that it's immobilized, and fibrinogen prefers to bind to heparin that's actually in solution. But you can see that TLT1 actually likes to bind to the immobilized heparin really well. So the dark blue line is also higher than the green line, and the dark blue line represents TLT1 that was made in a buffer called PBS, and the green line represents TLT1 that was made in a buffer called TIRODS. And so since the blue line had higher absorbances, that showed that the TIRODS buffer actually interfered with the binding between TLT1 and heparin. And this is probably just because TIRODS buffer contains some metal ions, and those typically do alter the binding. So then over to panel B. This just compares the two different buffers that were used with TLT1, and these are plotted as the TLT1 concentration on the x-axis, and then the average absorbance divided by the maximum absorbance. And this is a way of quantitatively finding a number that represents how well each sample bound to the heparin on the plate. So the number that you find is called the KD, and when I calculated it, the KD for TLT1 and PBS was lower than in TIRODS. So the lower the KD means that it bound tighter. So that shows again that TLT1 and PBS just bound better to the immobilized heparin. So these are more plate assays that I did with the heparin-coated plates. I'll talk about the one on the left first. So this one was testing a lot of different things, but mostly we just wanted to know if the imidazole was interfering with the binding, because the TLT1 that we use actually has a special tag on it that is used to purify it. And so if imidazole was altering the binding, that would show that it was actually just that tag binding to the heparin and not our actual protein. So we wanted to check that, and luckily the imidazole didn't really interfere with the binding, so that shows that it was actually our protein binding with heparin and not just the tag. And then we also tested a set of samples with heparin in solution that was combined with the TLT1. And the idea here is that the heparin in solution would compete with the heparin that's stuck to the plate for the binding of TLT1. So if this happened, we should see a lower absorbance of that sample, but the green line shows that we didn't see that happen. So that means that TLT1 actually prefers to bind to the immobilized heparin that's stuck to the plate, which is actually really interesting, and we didn't expect that to happen. And then we also tested sodium chloride and fibrinogen on these plates, and you can see that both of those interrupted the binding of TLT1 and heparin. And then over to the right graph, this one was also a plate assay with heparin-coated plates. I did a lot of those over the summer. So for this one, we just wanted to double check the different buffers for TLT1 because we were using slightly different concentrations, but you can see that the PBS still had way higher absorbances. And we also tested mannose on this one, and it didn't really interfere with the binding at all. But something that we found that was really interesting was that chondroitin sulfate actually reduced the binding of TLT1 and heparin quite a bit. And this was not something that we expected because chondroitin sulfate is in the same structural family as heparin. It's also a glycosaminoglycan. And so since we saw on the left graph that heparin didn't make any difference in the binding, we didn't really expect chondroitin sulfate to make a difference either. So that was really interesting that we found that. Okay, and these are Western blots that I did over the summer, and they just show proteins that have been run on a gel that separates them based on their size. And then you transfer those proteins onto a special paper called nitrocellulose. And so for this assay, I used heparin agarose beads, and those were bound to the TLT1 in the presence and in the absence of heparin in solution. So the TLT1 is the second set of bands from the bottom, around the 20 kilodalton mark. And the first two columns of those are the input samples. So these were just, they weren't put through the binding process. It's just there to show that your initial sample actually did have protein in it, just in case nothing shows up in your bound samples. And then the second two columns are the unbound samples. They were put through the binding process, but they just didn't stick to the beads. And then the last two columns are the ones that actually care about, really, and they're the bound sample of TLT1 that actually did stick to those beads. And then you can see that the very last row or column, the very last sample doesn't show up any less than the sample with no heparin. So that shows that the heparin in solution actually didn't interfere with TLT1 sticking to the heparin agarose beads. So that kind of just confirms what we also saw on the plates. And then we have those very bottom set of bands below the 15 kilodalton mark. And those are just lysozyme controls that you put in to make sure that protein could have showed up in your sample, and they're put in at a certain concentration, so you have something to compare your sample to. So for the Westerns, you can actually use a software that quantitatively measures how much protein shows up in each sample. So I use a software called ImageJ, and here's kind of the process of that and the results that I got. So the picture of the Western is how you start, and you basically just put boxes around your samples, and then the software can plot the signal of that sample, which you can see on the far right there. And then the software also calculates the area under each curve for you, and so that's what you see on the bottom. And then you basically just input those numbers into an Excel sheet and pretty much just divide the signal of your sample by the signal of the input, and that gives you the relative density of protein in each of your samples. So the chart on the bottom shows the relative densities that I got for the bound samples of each of the gels that I did, and then I just averaged them and got a standard deviation. And then I ran a statistical t-test to see if the samples with heparin had a statistical significant difference than the samples without heparin. So I got a p-value of 0.85. So anything above 0.05 for a t-test shows that it's not significant. So my samples definitely weren't significantly different, which just like it confirms what we saw on the plates, because this shows that there was no difference when heparin was added in solution. So that just further confirms that TLT1 actually prefers to bind to immobilized heparin.
Can y'all still hear me? Okay, cool. So Julia used her methods were more with immobilized heparin, and so the ones that I used with heparin in solution were fluorescence spectroscopy and electrophoretic mobility shift assays, or EMSAs. And starting off here with fluorescence, this is a lot, so if you'll just kind of focus in on A. What I did is I took heparin with a fluorescent probe attached to it, known as FITC heparin, and I monitored its changes in fluorescence when titrating in a protein of interest. And so what I did is I plotted its average relative fluorescence units versus its wavelength, and I excited FITC heparin at 490 nanometers. And if you'll recall from Gen Chem, you excite it, those electrons go up to a higher energy state. As they fall back down, I can capture their emission spectra from 510 to 550 nanometers. And FITC heparin does have a maximum fluorescence at 520 nanometers, which is important over here whenever we get to it. But I have this baseline of FITC heparin alone, and then I titrate in the protein of interest, in this case fibrinogen, in increasing concentrations. And if I see a decrease in fluorescence when I'm adding in the protein, that suggests that those two things are binding. And so the thought process behind that is that FITC heparin alone, its fluorescent probe is pretty well exposed, and it's going to have a high fluorescence. And then if I add in a protein and it's binding to heparin, it's going to cover up that probe and it's going to decrease its fluorescence. And so if those two are really binding, when I add in higher concentrations of the protein, it should continue to bind until it eventually saturates the heparin. And that is exactly what we saw over here in B. I plotted the maximum fluorescence of FTSE heparin versus the concentration of fibrinogen that I was adding in. And I see a drastic decrease in fluorescence until it eventually becomes saturated. So that suggests fibrinogen is binding to FTSE heparin in solution, which is exactly what Julia saw. And to also confirm that TLT1 is not really binding to heparin in solution, I repeated this with TLT1, and as you can see, with even higher concentrations of TLT1, we're really not seeing that drastic decrease in fluorescence. So with all of this, we get that fluorescence spectroscopy is a useful method to study fibrinogen and FTSE heparin binding, but not so much for TLT1. So more of Julia's methods are better to study TLT1 and heparin binding. And I did all of this against a buffer control here in E and F, just to make sure that these changes that I'm seeing are really due to fibrinogen and heparin binding, not just because of the buffer that it's in. And then I took this a step farther. So in plot A right here, this is exactly what you just saw. I'm just taking FTSE heparin, titrating in fibrinogen, but then I repeated it by adding in unlabeled heparin to see if it competed away for binding. And we thought, well, maybe since it's still heparin in solution, it should compete away for binding. We didn't see that. So here in the blue, it's just regular FTSE heparin titrating in fibrinogen. And then in the orange right here is adding in that unlabeled heparin to hopefully compete it away. But there's not a big enough difference between the two, between the orange and blue, to really say that in solution heparin is competing away for FTSE heparin binding. So we still need to do some stuff with this to see what the story is. And one of possibly like an issue with fluorescence spectroscopy is whenever you attach a fluorescent probe to a molecule, you can possibly alter its conformation so that the data that we were seeing before with fibrinogen and FTSE heparin, could it be that heparin and fibrinogen are really binding, or could it just be a result of the fluorescent probe that we attached to heparin? And in a way to make sure that heparin and fibrinogen are really binding, I can synthesize a fluorescent probe to put on fibrinogen instead, and then repeat those assays with unlabeled heparin and do it that way, just to make sure that what we're seeing is really a result of heparin and fibrinogen binding. And so that's what you're seeing right here is I've been synthesizing a fluorescent probe to put on fibrinogen, and this is a picture of it right here, super cute. I love when science has colors, it's very pleasing. But this synthesis project has been interesting. It's been a lot of piecing together different literature with the help of Dr. Duncan, because there's no like one literature piece that has the synthesis pathway for me to use. So there's been a lot of piecing stuff together, trying to figure out how to buy a reasonable amount of reagents, not use a lot of stuff. And it's also been a lot of safety training. So a lot of what I use is a respiratory irritant, or it's hazardous, toxic. And so I've had to wear this summer like a mask, a face mask, a respirator, my glasses and goggles. So just a lot of safety gear. And also just a lot of hoping that this works, because sometimes, well, a lot of times what you do in the lab just doesn't work. But after I synthesized my product, that what you just saw in the last picture, I wanted to make sure that what I synthesized was actually what I intended to make. And how I do that is I first check its melting point. And so if you see in this box right here, I put it in a capillary tube, like a little capillary tube, and then put it in a machine that can calculate its melting point. And what I got was 236 degrees Celsius, which was perfectly in the range of the literature value. So that gives me a rough estimate that what I synthesized was what I needed to make. And then I analyze it by putting it into a proton NMR spectroscopy. And that's nuclear magnetic resonance. So the easiest way to think of NMR is like it's a baby MRI machine. So I put my product in and I dilute it into a solvent, into just some liquid that doesn't interfere with the NMR, and I put it in there and a magnetic field is generated. And those atomic nuclei spin in a way that's characteristic and allows me to determine the structure of whatever I put in there. And so that's what I did is I took the data that the NMR gave me and I tried to piece together the structure of whatever was in there. And thankfully, I did determine that what I synthesized is what I needed to make. So if you look at these numbers right here, what's on the x-axis is very, very similar to what the literature gave me. But if you realize I'm not seeing that hydrogen over here at 11.1 ppm. And my thoughts on that are because, well, what I used, first off, I didn't make a lot of, so the solution was pretty dilute and it just might not have shown up. Or the literature source right here, they used an NMR instrument that had a much higher frequency than ours does. Ours is still great, it's going to do whatever you need it to do, but maybe that difference in frequency just didn't allow the hydrogen to show up. And so moving away from fluorescence, another method that I used was electrophoretic mobility shift assays, or EMSAs. And these methods are really nice to use because you don't have to go into a lot of quantitative analysis. You can just really visualize binding happening on a gel. So like Julia said, with gel electrophoresis, you can put in samples and they'll be separated by size. Smaller stuff will migrate towards the bottom. Larger stuff has a harder time migrating through the gel and will be up towards the top. And so if you add in a protein with heparin and they bind, they're going to form a larger complex that's going to stay more up towards the top. And in order to do a lot of these big tests, if you will, I first need to optimize the concentration of the protein and heparin that I'm going to use. And that's what you see here in these gels. I'm optimizing the concentration of fibrinogen to use. And I'd already optimized the FITC heparin, so I'm just using what I need to use. But I loaded in the standards so I could quantify these complexes. And then I took 30 mg per mil fibrinogen and I decreased its concentration down by 3. So you're seeing 30, then 10, then 3 mg per mil. And I did that without FITC heparin and then in the presence of FITC heparin. And so if you see right here, you can maybe see that some of that is still in the wells. And that's because whenever I was using higher concentrations of fibrinogen, there was a lot of solubility issues. Things are sticking in the wells. But when I got down to around 3 or 1 mg per mil fibrinogen, I wasn't having those same solubility issues, and I was still able to see the complex of fibrinogen and FITC heparin. And Dr. Gibson brought this up. It was really interesting to see that that was the optimal concentration range, because that's comparable to the fibrinogen concentration in the human body, around 3 mg per mil. So it was cool to just put that into context and see that it really does relate to the real world, if that makes sense. And the only difference between A and B and C and D are in A and B, I used phosphate buffered saline or PBS buffer. And in C and D, I used Tyrode's buffer, like Julia said, has some different metal ions in it, just to see if buffers have any effect in what I'm seeing. And like in fluorescence, took it a step farther, added in unlabeled heparin in solution just to see if that computes away for binding. And I did it in increasing concentrations based off of FITC heparin. And if you can see, it's kind of hard to see. There is a little bit of a difference in that formation, in that complex formation between fibrinogen and FITC heparin. But I need to continue to repeat this and do it in some different buffers just to make sure, just to get reliable data that's also repeatable. And before I explain this figure, I just want to shout out to Ariana Brown. Ari, thank you so much for helping me run this gel. She's starting to get in lab experiences, as well as Finn. Both of them are awesome. They're really excited to get into the lab, and they've been a big help with what me, Julia, and Caleb Finnegan have been doing in the lab. But here with this gel, S cubed, you all did an EMSA for your summer experience, and the standards didn't really show up. And so we thought maybe the standards were degraded, maybe it was the gel. We didn't really know what was going on. And so a couple weeks later, I ran some more gels with just standards, and the same thing happened. Nothing showed up. So we thought maybe it's the dilution of the stain, the DNA stain that stains the bases within the standard. And so I tried that. I honestly just picked some dilutions that sounded good to me, from 1 to 100 to 1 to 10,000. And I did that for 3 different standards just to see what standard would be best to use on the gels. And we saw that the dilution that S cubed was using at 1 to 10,000 really just doesn't show up well, and especially doesn't show up well after you stain the gel. So that's what the issue was. And I also optimized the concentration to use 1 to 1,000, and then also use both of these standards so I can quantify both the fibrinogen bands and the heparin band. And we put this in here just because it really is, we thought it was a testament to what we've done this summer and also just for the past few years of stuff doesn't work and you're like, okay, so now what? And you keep going off this process of finding answers to your questions, problem solving, applying critical thinking, and just doing a lot of things to get to the point where you can finally study what you want to study. It's a lot of background work. But with that, that is all of the figures and results that we've had talking about our methods. But we also wanted to talk to you all about what we liked and what we didn't like about working in the lab this summer, and really just in general. The first part of this, so the likes are on the left and the dislikes are on the right. At least for us, the likes were that it is science. You know, we are majoring in STEM for a reason. We enjoy science, and it was just nice to tangibly work with that and to work with our hands and be able to apply that knowledge to our classes. So that was just one thing that we liked about working in the lab this summer.
Another thing that's really good is that we kind of get to set our own schedule, because we've kind of graduated to the point where we can work pretty independently in the lab, so we're able to just go in at convenient times, which is pretty nice. But we also get to work with other people that are in the labs. There's like, I think 5 students now. We're collecting new ones. And so they are there, like you can just support each other when you're learning new things or when your experiments are not going well, or you can just talk through an experiment with someone else in the lab, which is really nice.
Like I said before, it's interesting to see how your critical thinking and problem solving skills develop over the time of working in the lab, because when you first come in, it's really just tell me what to do and I'll do it. And like Julia said, now we're at the point where we can do things independently. We can figure out why something didn't work and then do another study to answer that question. And it really is just nice to see all the time and effort that you put into working in the lab finally pay off and be to a point where you're like, I can do this. I don't have to have someone tell me everything to do.
Yeah, it's definitely rewarding when you've worked really hard on a certain experiment, like you've had to repeat it several times and it finally works at the end. That's really a good feeling to have when you've figured out what went wrong all the other times. And yeah, working in the lab has definitely taught us problem solving skills, because a lot of times you have to adjust your experiment in the middle, or you have to adjust it for things that you didn't even think would be a factor in it. So that's just really helped us think a lot more critically about things.
So on the flip side of that, some of our dislikes are that, for one, you are inside a lot, especially if you're doing it for like a job, like Julie was for the Leyford. You're inside a lot, and Sutton does not have any windows, so you're just kind of looking through the one window and wanting to be outside. And sometimes when things don't work, or when you're just in there for a long day, it can be stressful, and sometimes you can just get mentally exhausted. So it's really been important for us over these couple years to just take breaks for yourself, you know, practice self-care and just say, you know, I have maybe 10 hours of work to do this week, but maybe I'm going to split it in 5 hours this week, 5 hours next week, or just take a week off because I just need to focus on classes or something else. So it can be stressful, but if you have that teamwork and support to kind of talk it out and also take care of yourself.
Yeah, another not very great thing about working in the lab for several hours at a time, sometimes you can like forget to eat a lot, or you just don't have time, so that's not really great. But you figure out when you can take breaks and when you can have a minute to step outside and go eat lunch. And then a lot of the time things don't work out the way that you originally wanted them to, and you work really hard on this whole long experiment, and then you get the results and it doesn't work. And so that's kind of a bummer, but you just have to kind of look forward to when it does finally work, and you have to work through all of the problems, figure it out, and then it's just rewarding when it finally does work. So our future plans, Our immediate future plans are just to finish our thesis this semester. And then after that, we're hoping to combine our theses and publish something, which would be really great. And then we also plan to just keep gaining experience by finishing thesis and probably working in the lab even after we're done with thesis, just to help get more experience for our future careers. And then we're also hoping to present our stuff at a conference, but you know, with COVID, everything is getting canceled right now, so that's kind of uncertain on if we're going to get to do that, but we're really hoping that we'll be able to. And then hopefully we will both be going to graduate school in the next year. Yeah, so I think that doing this research has definitely prepared us for that, and I think it's really going to contribute to our future life in grad school and in our careers.
So some advice that we've tried to piece together for y'all is just talk to your professors, because since we're such a small school, you really do have that personable and professional relationship with your professors, and they are just a Zoom call away, whatever. So don't be afraid to talk to them, and, you know, ask any questions that you may have, and, you know, ask about their projects that they're doing, because each professor in the STEM in Sutton essentially has something that they're doing, and they would love to have you on and work, you know, on their project. And also talk to your peers. Like we said, Boomer Russell is kind of the reason we're here, and we started so early. So don't be afraid to talk to your, you know, people who are already, you know, your juniors and seniors who are involved. Don't be afraid to talk to them, and don't be afraid to try things out, even if you have doubts about it. Like, I mean, you can always say no, and you can always stop doing it. So it's beneficial to try it out. And also when applying for scholarships, applying for grants, applying for really anything like an REU, an internship, don't be afraid to apply, because again, the worst thing that they're going to say is no, and there's always more things that you can apply for. And we were trying to find a nicer way to put this, but really what we came up with was just roll with the punches. Especially, that's kind of like the theme of 2020 at this point. You just got to keep moving forward and try to have some hopeful attitude about you that it's going to get better, or, you know, whatever you can do. Just roll with the punches and we'll make it. And with that, that concludes our presentation, and we'd be happy to take any questions.
I have a question. Okay, so how did, what made you guys like realize that you wanted to study this in particular?
Julia, do you want to start it off, or me?
Well, it was kind of just something that Dr. Gibson was interested in, and when we started in the lab, we were just kind of doing bits and pieces of other people's projects, and then we kind of just wanted, this was kind of the one that hadn't been explored to the fullest extent, so we just wanted to kind of take it and figure out what we could about it.
Yeah, and for me, it was the same thing. I just kind of wanted to check it out. I just wanted to get the lab experience, but then the more I was in it, the more interested I became in it, and just kind of took it on as my own. And yeah, so I guess it just kind of started out, and then we were like, oh, this is really cool.
Okay, thank you.
Guys, I like the way that they highlighted that you start early and become involved with the professors, and that you ask questions, you mess up with stuff, it's okay, because I think that that is really important, especially at this point when we're getting ready to declare majors and we're trying to think about what we want to do. It's really like, you can get out there and ask questions, and they talked about how easy that is to do. So
Boomer, did you have a question for us?
Why do you have to call me out like that? But I do. I'm curious about it was in Julia's section about the chondroitin sulfate and the mannose.
Mm-hmm.
Why did you choose those two in particular to test? Is it a structural thing, a charge thing?
Well, for the chondroitin sulfate, that was just the only other glycosaminoglycan that we had in the lab.
I feel that.
So that, but we're trying to do it with also hyaluronic acid. So we're gonna test more of them. But yeah, it was basically just because that's what we had on hand at the time.
I feel that. Thank you.
You're welcome.
And you all did a great job.
Thanks.
Okay, any other questions? Jordan, will you go ahead and flip to the QR code? Okay, guys, if you will scan this, it will bring up a short survey, and you just fill those out, answer them, and that's what you need to fill out for the survey.
Lindsay, can we also put a link to that in the chat in case people want to go directly with their computer? Is that possible?
Maybe.
Or we can, you know what we can do? We can send it to the participants. We have everybody.
Yeah, we'll send it out to
There we
the
go.
people. Okay. I'll email it out to everyone who's here.
Oh,
What did?
it has reached its scan limit. Do I have that link in my email?
Yeah, I think so. I think I sent the link along with the code scan, or the code, I mean.
Well, I think we might have to send it out.
Yeah, that's no problem. I can send it to the participants. Oh, there it is.
Somebody put it in for, oh, thank you.
Yeah, so I just put it in. The link in the chat should work for you. Please let us know if it doesn't. We'll email it out also. And we thank you very much for filling this out for us. It's helpful for the whole program so that we can figure out what we are learning. So thank you. And great job. Can I just say I'm so proud of my people. Yay, really great job. So proud of them. And thanks to Boomer for teaching them everything they know. And honestly, I'm at a point now, I feel obsolete because like all my students know how to do things that I don't know how to do now because they've learned from each other. You know, Boomer learned something from some videos or whatever, and he taught the students, and now I don't know. So in many, many ways, my students are smarter and more skilled than me. And that's a hard thing to admit as a professor, but that is what we actually are aiming to do here is to teach our students how to learn and grow beyond where we are. So, yeah, so I'm so proud of these guys. And so thank you all for doing such a great job.
Yeah, and
Thank you.
to echo just one more time, like it's amazing that they have the opportunity to be independent in these labs. Because, you know, at bigger places, that's not always the case until you're at the graduate level. And so it's actually really awesome that they are saying, you know, hey, we learned these things, we are comfortable in the lab, we can go in whenever. And these are like your peers that are doing these experiments. And so I think that's pretty cool. And I guess with that, as long as you fill out the survey, which is in the chat, but I will also email, we thank our presenters. You guys are awesome.
Woo-woo.
And that is the end of our first presentation.
Can we put a small plug in for the next one? The next one is when?
The next one is October 6th.
Oh, can I say one thing? Oh, sorry.
Yeah.
I was going to say, if any of you are interested in working in Dr. Gibson's lab, just getting some experience, you can always email me and Julia and Dr. Gibson and even Kaylin Finnegan if you want to get plugged in. Just want to put that out there.
Awesome. Thank you all for being here and for your attention.
Thank you.
Is it October 6th at the same time?
Yes.
Thank you.
So all the presentations will be at the same time at 12:25. They'll all start then. And they'll all also be recorded, and then they'll be eventually uploaded to the YouTube channel as well.
Will it be the same code, or will we get a different code?
It's a
For different
different link every time.
Okay.
So each thing has its own link, which I'll